Alright, these are going to be the lecture notes over the citric acid cycle from the professor. I'm just going to go through what I've written out. These are the ones that the professor highlighted, the most important information of each of the slides instead of going through the whole thing. Alright, so the citric acid cycle happens after we've made our acetyl-CoA in the mitochondrial matrix. Now we're going to be in the mitochondria again, but we're just going to be processing that acetyl-CoA. And so 1 turn per glucose, this is going to be what's made in the citric acid cycle. 6 NADH, 2 FADH, 2 ATP, 4 CO2, and of course there's going to be the 6 hydrogens, but these are the 4 main things. And that's again for per turn per glucose. So 6 NADH, 2 FADH2, 2 ATP, and 4 CO2 per turn per glucose. Alright, so acetate, so step 1, this is just like a rundown. Step 1, acetate, which is a 2-carbon molecule from our acetyl-CoA, plus oxaloacetate, which is a 4-carbon. We're going to make citrate using the citrate synthase. We're going to make a carbon-carbon bond. Step 2, dehydration and rehydration will create isomerization, and we're going to get to our isocitrate. We're going to be using an aconitase, that's the enzyme in step 2. Step 3, we're going to have some decarboxylation. This oxidative decarboxylation will give us NADH plus CO2, and then the product for step 3 is alpha-ketoglutarate. Step 4, we're going to have another oxy-decarboxylation. Let me zoom in here. An oxy-decarboxylation in step 4 will give us NADH again plus CO2 and succinyl-CoA as the product for step 4. This involves the alpha-ketoglutarate dehydrogenase complex, I believe, for step 4. Let me see. Yeah, this is the complex again, alpha-ketoglutarate dehydrogenase complex. Alright, step 5, we're going to have some phosphorylation. We're going to actually have this substrate level phosphorylation. Step 5, we're going to be taking the succinyl-CoA. We're going to be using succinyl-CoA synthetase. So this is a synthetase. I spelled that incorrectly, I think. Synthetase. Yeah, I added an extra T. Or an extra H. There we go. So, succinyl-CoA plus the succinyl-CoA synthetase, we're also going to need we're going to make CoAsh, and we also need GTP. So this succinyl-CoA synthetase in step 5, it's going to actually make our GTP. But GTP could be converted into ATP, so I just wrote ATP there. The product of step 5 is succinate. So step 6, we're going to have dehydrogenation of the succinate, and we're going to get FADH2 plus fumarate. Fumarate will be the product of step 6. The enzyme used is, let me find it, sorry about that. The enzyme used is the succinate dehydrogenation. And step 7, we have a hydration reaction. That fumarate is going to be hydrated by fumarase. And we're going to, of course, need H2O in step 7. And we're going to be forming the product L-malate. This final step here, step 8, is a dehydrogenation. And this is going to involve malate, L-malate dehydrogenase. And we're going to be forming our third NADH, and more importantly, we're going to make our oxaloacetate. We're going to regenerate the oxaloacetate. And then you could go through the cycle again and again and again. Alright, let's go in depth with the steps. I know I usually make a separate video for this, but we kind of have it here and a lot of good parts into it too. Okay, so step 1, again, that's the carbon-carbon bond formation. We're going to take oxaloacetate and acetyl-CoA. We're going to have our co-ash come off of this, and we're going to be using that citrate synthase, not synthetase, but synthase. We're going to be making our citrate as the product. This is going to be acid-base catalysis. This is rate limiting. This is irreversible, so therefore we could talk about regulation with it. The substrate amount and the product inhibition are the ways it can be regulated. About this citrate synthase, this enzyme that's used in step 1, we have this induced fit property. So this induced fit property will stop the unnecessary hydrolysis of the acetyl-CoA thioester. You don't want that to happen. So the conformational change will happen when it's bound to oxaloacetate. When it's closed, the oxaloacetate will cause binding site for acetyl-CoA, and it will protect the carbanion. So the closed state, that's when the binding of oxaloacetate creates a binding area for acetyl-CoA. So our citrate, so step 2, citrate from the previous one step, we're going to use aconitase, and then remember this is dehydration then rehydration. And the aconitase enzyme will make R-isocitrate, so citrate to R-isocitrate. Removing H2O will create this cis carbon double bond to another carbon. The pore leaving citrate, the pore leaving group, which is citrate, will become a good leaving group as a secondary alcohol. This R-isocitrate is a secondary alcohol. The secondary alcohol is a good substrate for oxidation. There's an iron sulfur center in our aconitase. This iron sulfur center is sensitive to oxidative stress, and you need the amino acid cysteine for it. Cysteine, again, has those sulfurs in it, so those sulfurs are very important. Again, it's the iron sulfur center. Step 3, we have our isocitrate, our R-isocitrate. We're going to be using the isocitrate dehydrogenase to do oxidative decarboxylation. When we do this, let me see what we're going to be making. We're going to be making our NADH. We're going to also be making the product alpha ketoglutarate, and we're also going to make CO2 from that dehydrogenase. Step 4, we're going to take our alpha ketoglutarate from the step 3. We're going to do another oxidative decarboxylation. This is the final time in the citric acid cycle this will happen. We need the 5 coenzymes from our PDH complex, because now we're going to be dealing with the alpha ketoglutarate dehydrogenase complex. Again, it's the same type of deal here. There are 3 enzymes, and we need those 5 coenzymes at step 4 again. The complex, the AKG-DH complex for alpha ketoglutarate, when the enzyme is doing its job, it's going to make our product acetyl-CoA and CO2. We're going to have that CO2. Alright, there's going to be a different active site in this complex, however, but after 2 turns, we have the net full oxidation of glucose. The lost carbons are from oxaloacetate. These CO2s are from oxaloacetate, not acetate. Again, the 2 CO2s. So again, in step 3, we made a CO2 using isocitrate and the isocitrate dehydrogenase. In step 4, we made another CO2 from alpha ketoglutarate and the dehydrogenase complex. Those are from oxaloacetate. Those 2 CO2s are from oxaloacetate, not acetate. Step 4 is also irreversible, so we could regulate it. You see if there's anything else. This H2C, the CH2 to COOH, is from glucose. That's the thing that doesn't change. That's the part of the molecule that doesn't change. Those are from glucose. I should probably draw that. So this molecule, so like from citrate down, you have this H2C COOH, this portion right here, and then you have COH, and then COOH, and then another one here, H2C COOH. This part, going forward in the reaction, this came from glucose. This doesn't change. The CO2s, however, came from oxaloacetate. All right, step 5. This is where we're going to have that oxidative phosphorylation. Oh, no, sorry. Not oxidative phosphorylation. We're going to have substrate level phosphorylation at step 5. Pardon my mistake there. We're going to take our succinyl-CoA from step 4. We're going to be using a synthetase this time. So we're going to need GTP, and we're also going to make CoAsh. We're going to have this succinate as our product from succinyl-CoA with the succinyl-CoA synthetase. We're going to make succinate and GTP. GTP, again, could be converted to ATP easily. All right, so this is going to have the histidine phosphorylated in order for this enzyme to do its job, it looks like. Let me see. Wait, is that correct? Let's see. The histidine is there for step 5. Yeah, so do agree that the histidine is there. That's important. Let me see if we actually need it phosphorylated or not. No, I have that wrong. The histidine is phosphorylated, and this looks like it. This is what causes the GTP to be made. It doesn't look like the first part of it where the succinate is made. The histidine is not phosphorylated, but histidine is phosphorylated, and then that phosphate is transferred to the GTP to form it. All right, step 6, we're going to take our succinate. We're going to have our FAD and our succinate dehydrogenase, and we're going to be forming fumarate as well as FADH2. This is a redox reaction where we're taking the alkane, the succinate, into an alkene, the fumarate. This is going to be in the mitochondria inner membrane, not the mitochondrial matrix. This is going to be in the inner membrane where it's actually complex 2 in the electron transport chain as well. So this succinate dehydrogenase is also complex 2. There's a low reduction potential of C bound to H, so therefore we can't use NADH for this step, so we have to use FAD. This FAD is actually covalently bound, and that's very strange. This is very strange. Step 6, the succinate dehydrogenase's FAD is covalently bound, and step 6's reaction is very close to equilibrium, which is also interesting too. Also in step 6, we have this malonate. This is a molecule that looks like succinate, except for it has one less CH2 in its molecule. So succinate has 2 of these CH2s. Malonate only has 1. Malonate will block the TCA cycle. It's a competitive inhibitor of succinate dehydrogenase. All right, step 7, we have fumarate. We need H2O here and fumarase because this is the hydration reaction. This will be forming L-malate, not malonate, L-malate, M-A-L-A-T-E. This step 7 is reversible, and the forward reaction of this only works on the trans double bond. However, the reverse will do both. So in the reverse, we'll have fumarase acting equally stereospecific. We could have L-malate and D-malate being converted back to fumarate or malleate. However, fumarate, which is the trans double bond, that's the only molecule that will be worked on going in the forward direction. So fumarate is trans, then we'll have our fumarase and H2O, and then we'll make L-malate. Step 8, this is the final step. This is going to be our L-malate, which is an alcohol. we're going to be adding the NAD and the malate dehydrogenase, our L-malate dehydrogenase, and we're going to be forming oxaloacetate plus NAD. Oxaloacetate is a ketone. So we're going to, again, regenerate OAA for the whole cycle, and this is unfavorable, and reversible. This is unfavorable. We're going to use the Chatelier's principle to pull it forward. Also, this is the last time we make NADH in the cycle in step 8. This is a third NADH, but then since we're doing 1 turn per glucose, it's actually 6 NADH, 4 CO2, 2 FADH, and 2 ATP, because again, we have 2 molecules of acetyl-CoA. Alright, so again, back to that page. 1 turn per glucose, 6 NADH, 6 hydrogens, 4 CO2s, and 2 FADHs, as well as 2 ATP. If it was just acetyl per acetyl-CoA, that would be 3 NADH, 1 FADH2, 1 ATP, and 2 CO2. Again, we're just doubling it because glucose will split off into 3 carbon molecules. So there will be 2 of them. And then, 2 pyruvate, so 2 acetyl-CoA, and then so that's why we multiplied the numbers in the PowerPoint by 2. So 6 NADH, 4 CO2, 2 FADH2, and 2 ATP per turn per glucose. The whole reaction would be acetyl-CoA plus 3 NADH, or acetyl-CoA plus 3 NAD plus FAD plus GDP plus P plus 2H2O will make 2 CO2, 3 NADH plus GTP plus CoA plus 3H plus FADH2. That is crazy. Net oxidation of the 2 carbons to CO2, this is equivalent of 2 acetyl-CoA carbons, but not the same carbons, it's just the equivalent of. The energy captured by electron transfer to NADH and FADH2, that's where that's happening. Again, GTP, which would be converted to ATP. A large flow of electrons into the respiratory chain via the NADH and FADH2 will lead to the formation of almost 10 times more ATP during the oxidative phosphorylation, also known as the electron transport chain. NADH will get you 2.5 ATP, so you would multiply the amount of NADH by 2.5 ATP, and then FADH2, you'll multiply the amount of FADH2 by 1.5 ATP. In that respect, glycolysis plus Krebs will get you 30 to 32 ATP. Okay, so the citric acid cycle is an amphibolic pathway. That means it's both anabolic and catabolic at the same time. Animals, we can't do acetyl-CoA to glucose. We can't make that conversion. So therefore, there's no net conversion between acetyl-CoA and going into oxaloacetate. However, bacteria, plants, fungi, and protists, we could use this glyoxylate cycle where acetate will become carbohydrate. There's a difference there. Alright, so citrate can become fatty acids. Alpha-ketoglutarate will become glutamate, and then that becomes other amino acids or a purine. So acetyl-CoA could become porphyrins or hemes. Hemes are like in the hemoglobin, so acetyl-CoA will turn into some of those proteins. Oxaloacetate will become phosphoenolpyruvate as well as some amino acids. The reactions in the citric acid cycle, the intermediates, I should say, give it this term here, this anaplerotic. Anaplerotic means that the reactions are used to regenerate the intermediates. For example, pyruvate plus bicarbonate plus ATP with the pyruvate carboxylase will make oxaloacetate. Also, the phosphoenolpyruvate plus CO2 plus GGP plus our carboxykinase, our PEP carboxykinase, will make oxaloacetate. Those are like from gluconeogenesis. Regulation, so we've talked about the PDH complex in the previous video. Citrate synthase. Citrate synthase is inhibited by succinyl-CoA. Isocitrate dehydrogenase will control the levels of citrate. Citrate will then control the levels of the phosphofructokinase 1 in glycolysis. Citrate will leave the mitochondria to do that. And then the other part of regulation, alpha-ketoglutarate dehydrogenase complex acts in the same way as the PDH complex. Therefore, that means that when it's the presence of the kinase and it's phosphorylated, this means it's off. However, when we have our phosphatase and it's not phosphorylated, this means it's on. And then the normal things also drive it forward. Low ATP, high NAD, and high AMP will push the citric acid cycle forward. All right, thanks so much for tuning in. Please do something nice for someone and take care.